Today I am taking up the topic structure of the eye and the mechanism of vision. We have a pair of eyes situated in the sockets present in the skull. Each eye is nearly a spherical structure having three layers. From outside to inside, the first layer, the outermost layer is called sclera or white of the eye because of its whiteness. It is made of dense connective tissue. It is a tough layer, gives protection to the eyeball. At the anterior part of the eye, the sclera is transparent and it is known as cornea. It is a transparent window of the eye through which light enters the eye. You must have heard about people going blind because of the lack of transparency of cornea. If the tear secretion or lacrimal gland secretion is absent, cornea loses its transparency and becomes white, causing blindness. It's called corneal blindness. The corneal blindness can be removed by transplanting a healthy cornea from dead people. Cornea of any person can be given to any person. There will be no rejection because cornea is without any blood vessels. So the middle layer is known as choroid layer. It is pigmented and also contains blood vessels. Because of the pigmentation and presence of blood vessels, it appears bluish in color. The choroid layer is thin in the 2/3 of the eye, but at the anterior 1/3 part, it is a bit thick. And the choroid layer produces anteriorly ciliary bodies. These are the ciliary bodies. The ciliary bodies produce iris. This is iris. Iris is the pigmented structure covering the lens, eye lens. In the middle of this iris, there is an aperture known as pupil. So light entering the eye through cornea passes through the pupil and falls upon the lens. Then after falling upon the lens, it is transmitted into this chamber. The ciliary body secretes a type of fluid, a watery fluid known as aqueous humor. Then, this is the crystalline lens, biconvex crystalline lens, which focuses light on the retina. This lens is supported by or suspended by ligaments, suspensory ligaments arising from the ciliary body. The ciliary body has also a muscle known as ciliary muscle. I have told you this is the pigmented part of the ciliary body called iris. Iris has radial muscles. With the help of the radial muscles, the size of the pupil can be dilated or narrowed down. Then, the innermost layer is called the retina. It is a very important layer. It contains the photoreceptor cells. Photoreceptor cells, that means the cells that are sensitive to light. Coming to the photoreceptor cells, there are two types based upon their shape and also function. First type are known as rods. Second type, cones. Rods look like this. They got a rod shape. These are the cones. They look like a cone So based upon their shape, they are known as rods and cones. The arrangement of rods and cones we will see after some time. The rods are concerned with twilight vision or scotopic vision. That means dim light vision. When the light is low, these structures help in visualizing the things. The pigment present in the rods is known as rhodopsin. Rhodopsin. Rhodopsin is also known as visual purple, derived from vitamin A. Derived from vitamin A. So, vitamin A is very important for efficient functioning of rods. What happens if there is deficiency of vitamin A? It affects the functioning of the rods. And such people cannot see during dim light or in the dim light. They fail to see in the dim light. That is called night blindness. So, night blindness is due to deficiency of vitamin A. It is not a disease. It can be easily cured by giving vitamin A to such persons. But there is another eye problem or blindness known as color blindness. Color blindness is a genetic disorder transmitted by the X chromosome. So it is also known as a sex-linked disease or a recessive sex-linked disease. And there is no cure for color blindness. There is no cure for color blindness, but night blindness can be cured by giving vitamin A. Then, so this is the inner layer, retina. Here, the optic nerve enters the eye. Sorry, optic nerve leaves the eye and optic blood vessel enters the eye. This is opposite to this pole. So, here, where the optic nerve leaves the eye and optic blood vessel enters the eye, there are no photoreceptor cells here. Practically, no photoreceptor cells are present. This point is known as blind spot. No visual activity occurs in the blind spot because there are no photoreceptor cells. Just by the side of the photoreceptor cells, there is one area known as macula lutea. It's called macula lutea. It is enlarged here. Macula lutea is also known as yellow spot because of its color. In the middle of macula lutea, there is a depression known as fovea. The depression is called fovea. Fovea is packed with cone cells, these cells. So it appears yellowish in color. And in the fovea, vision is very acute or resolution is very high. So fovea is known for acute vision or high resolution. Then, again coming to the photoreceptor cells, sorry, before that I will take some other points. See, there are two chambers in the eye. There is one chamber in the front of the lens, another chamber behind the lens. The anterior chamber is a smaller one and this is a bigger one. The anterior chamber is known as aqueous chamber. The aqueous chamber is filled with a watery fluid known as aqueous humor. Aqueous humor. The aqueous humor is secreted by the ciliary body. The ciliary body secretes aqueous humor, and that is released into the aqueous chamber. Excessive aqueous humor is released into canal of Schlemm. Canal of Schlemm. And from the canal of Schlemm, the excess fluid is released into the venous system. So actually, canal of Schlemm is a part of the lymphatic system. So the excessive fluid is released into the lymphatic system, and from there it goes into the venous system. Then, the bigger chamber present behind the lens is known as vitreous chamber. It contains a jelly-like viscous fluid. It is jelly-like, very thick, but transparent. Otherwise, the light can't pass through. The vitreous chamber provides a definite shape to the eyeball. Otherwise, the vision will be affected. So, thus there are two chambers. Aqueous chamber in front of the lens and vitreous chamber behind the lens. Now, coming to the photoreceptor cells. Already I have told you, the rods have a rod-like shape. They have a visual pigment known as rhodopsin and derived from vitamin A. So in the retina, rods are arranged like this. This is the rod. Here we have to remember one very important point, because it leads to a lot of confusion here. So this is the vitreous chamber, let us say. This is the choroid layer. So this is the choroid layer, this is the vitreous layer, this is the retina. I am talking about this part. So this part is enlarged here, from here to. So the rods are just below the choroid layer. Similarly, the cones are also situated just below the choroid layer. Coming to the cones, these are concerned with photopic vision or daylight vision. So rods are concerned with dim light vision, whereas cones are concerned with daylight vision or photopic vision. Cones also serve another function. They are also concerned with color vision. We can see colors with the help of the cones. The cones are mainly three types. Red, some are concerned with red color, some are concerned with green color, some are concerned with blue color. These three colors are known as basic colors. By the combination of these three basic colors, millions of different color shades can be produced. So in nature, there are only three basic colors: red, green and blue. By differential combination of these colors, different shades can be produced. So, but one important point you have to remember: what happens if you focus the light of same intensity on all the three types of cones at the same time? Then what happens? When they are stimulated simultaneously with the same strength of light, white light is produced. They cannot produce any color. That we have to remember. Then, coming to the retinal wall. This is the retinal wall. In the retinal wall, we come across three types of cells, arranged from choroid layer towards the vitreous chamber. Immediately below the choroid layer, the photoreceptor cells are arranged. This is the shape of the cone, this is the shape of the rod. Each cell has two parts or two segments. This is called outer segment, this is called inner segment for cones. Similarly for rods, this is the outer segment, this is the inner segment. The photoreceptor pigment is present in the outer segment, whereas mitochondria are present in the inner segment. The mitochondria produce ATP, which is used in the transmission of action potentials generated in the photoreceptor cells. Because of this function, we find a large number of mitochondria in the inner segment of rods and cones. Then, this is the axon. This is the axon. So these are axon terminals, and they synapse with dendrites of bipolar cells. While dealing with the nerve cell, I have told you, there are bipolar cells in the retina of the eye. These are the bipolar cells. They have only two processes. This is the dendrite and this is the axon. This is the dendrite here, axon here. The bipolar cell receives signals through dendrite and sends them away through axon. Like this. These are axon terminals here and here. And below the bipolar cells, there are the ganglionic cells. Again, this is the synapse between the ganglionic cell and the bipolar cell. Then the ganglionic cells produce processes that form a part of the optic nerve. So in this way, an optic nerve is formed by the processes coming from the ganglionic cells. The optic nerve goes to the brain, and in the visual cortex of the eye, in the brain, in the visual cortex of the brain, an image is formed. An image is formed. What type of image is formed in the eye means, in the eye or in the retina, the image formed is small and inverted. So here, small and inverted image is formed. But in the brain, complete image is formed. Upright, complete image is formed in the visual cortex. Now, let us see the mechanism of vision. So, light enters the eye in this way. It passes through cornea, pupil, lens, and focuses on the macula lutea or on the retina, let us say. Here we have to remember one point. So this is the vitreous chamber, let us say. From here to here, retinal wall. So in the retinal wall, I have told you, from here to here, the arrangement is like this. Photoreceptor cells, bipolar cells, and ganglionic cells. Light entering the vitreous chamber travels through the retinal wall up to the choroid layer. So the light from the vitreous chamber, it travels to the entire width of the retina and enters the, it travels up to the level of choroid layer. As it travels, it falls upon the rods and cones, and excess light is absorbed by the choroid layer. The function of the choroid layer is to absorb excess light that is entering the retina. So when the light falls upon the rods and cones, what happens? It stimulates the photoreceptor pigment. In the case of rods, what happens? I have told you, in the rods there is a pigment known as rhodopsin. When light falls upon the rhodopsin, that means here, rhodopsin dissociates into two substances, opsin and retinol. Opsin is a protein, is a type of protein. Retinol is an aldehyde of vitamin A. So when the light strikes the rods, retinol dissociates into opsin rhodopsin dissociates into opsin and retinol. The opsin on dissociation undergoes structural changes. So the protein opsin undergoes structural changes on dissociation. These structural changes cause permeability of the cell membrane of the rods. Permeability of cell membrane of the rods, just like in the case of action potentials generated of a neuron. And gates open, sodium ions travel into the cell, and action potentials are generated. So, dissociation of opsin brings about structural changes, and these structural changes make the membrane permeable for ions, and the action potentials are generated in the rods and cones. In the cones, the different pigments are there. In the rods, rhodopsin is there. So once the action potential is generated, you know that it will not remain stationary. It travels throughout the length of the neuron. So action potential generated here, they travel all along the length of the axon. This is the axon, and they reach the synaptic junction present between bipolar cell and the rod. They cross the synaptic junction, pass through the bipolar cell. Again, they cross the synaptic junction here. Then they enter the ganglion cells, and through these branches of optic nerve and optic nerve, they reach the visual cortex of the brain, where complete and upright image is formed. In that way, the vision, the mechanism of vision is present in the human beings. So in a nutshell, this is about the structure of the eye and mechanism of vision in human beings. Thank you.